Okay, good morning or good afternoon, wherever you are in the world. Welcome on behalf of ADVAC to this new webinar on the safety of COVID-19 vaccines. I mean, clearly a topic of great interest, and we had many of you registering for this webinar. Clearly, as there will be many of you, we need to be orderly for the question period, and you can send your question in writing to the chat box, copying everybody, and our chair will monitor the questions and invite you in turn to ask your question live. So be ready to activate your video when you want to ask your question. I will, without any more delay, then turn to our chair. Melinda Wharton was kindly accepted to chair this session. Melinda doesn't need to be introduced, I guess. She had such a long career at CDC in immunization delivery, and now she's the Associate Director for Vaccine Policy, and she's been for many years part of the ADVAC Scientific Committee. So I will leave you in her capable hands. Thank you.
Thanks so much, Philippe, and welcome to all of you. I'm so glad you were able to join us today. This has been an extraordinary last couple of years in the vaccine world, with very rapid development of multiple vaccines for prevention of SARS-CoV-2 and global implementation with many hundreds of millions of doses of different vaccines administered in a very short period of time. It's been a demanding period for our vaccine safety systems globally, and I think those systems have served us well in terms of identifying safety issues and providing risk mitigation as we can. So I'm really delighted to have two expert speakers today who can share some of the COVID-19 vaccine safety experience with you. We have two speakers. Our first speaker is Dr. Rita Helfand, who is chair of the WHO's Global Advisory Committee on Vaccine Safety. She also is a senior advisor for science in the National Center for Emerging and Zoonotic Infectious Diseases at CDC. She joined CDC in 1992 as an Epidemic Intelligence Service officer. She received her MD from Harvard Medical School and completed her pediatrics training at Boston Children's Hospital. Our second speaker is Dr. Tom Shimabukuro, who is the former Deputy Director of the Immunization Safety Office and the current Acting Director of Immunization Safety at CDC. He also has served as team lead for the vaccine safety team in CDC's COVID-19 vaccine task force. Dr. Shimabukuro received his MD degree from New York University and did his internship at Emory University School of Medicine and his preventive medicine residency at Johns Hopkins Bloomberg School of Public Health. He also completed the Epidemic Intelligence Service program at CDC. And with that, I will turn things over to Dr. Helfand for her remarks.
Thank you very much, Melinda. And for the first chance, good morning, good afternoon, good evening, everybody. And thank you for allowing me to speak. My first challenge will be to see whether I can get the screen up and running. So fingers crossed. Okay, it looks like I'm up. Melinda, can you?
Yeah, it's good. It's good.
Great. Thank you. So as Melinda said, I'm going to be speaking on global vaccine safety from the perspective of the WHO Global Advisory Committee on Vaccine Safety, or GACS, and they created a GACS COVID-19 subcommittee, and I'll get into that more. I'm going to be giving you that perspective, and then Tom will be following me, following on some of the same areas, but from a country perspective, so you can see how these things interplay. I have nothing to disclose. I realize that most of you already know a lot about the vaccine safety basics, but I do want to make sure that we're all starting from the same page. So I'm just going to run through some of that very quickly. As we all know, vaccines are different than drugs, and there's a higher standard of safety that is expected. That vaccinees are generally healthy, and somebody who's ill, for instance, with cancer, is going to be much more willing to accept a serious adverse event if it means that they can potentially cure their cancer. Vaccines also serve the dual role of both individual and societal protection. The level of risk that is tolerated is going to depend on a number of factors. The risk and severity of a vaccine adverse event. For instance, somebody is going to be much more likely to tolerate a sore arm than they are a blood clot. The risk and severity of the vaccine preventable disease, and whether there are alternatives to either an alternative vaccine or therapeutic. What have we learned from the past about what might be an acceptable risk? It's very hard to generalize, but if you think about Guillain-Barré and that some years there's a potential association with the flu vaccine, people seem to accept a potential attributable risk of about 1 in a million for that. I realize that everybody here is very familiar with the vaccine safety life cycle, and the main reason I put this up here is to clarify that, remind us that by the time you've gone through your phase 1, 2, and 3 trials, you have a pretty good sense of how the vaccine's working as you've given it to tens of thousands of people. But we do still need to do the post-authorization or post-licensure monitoring to be able to identify those new or rare events, changes in patterns of the known events, identify any safety problems in special populations such as pregnant women, the elderly, look for risk factors for particular adverse events, and then assess the safety of vaccine lots, which I will not be talking about the last. We typically talk about 2 different types of adverse events following immunization, or AEFIs, and adverse events of special interest, or AESIs. For the AEFIs, these are any untoward medical occurrence that occurs basically just sometime after being vaccinated. So it doesn't have to be causally associated with the vaccine. For instance, you could develop an influenza illness after you got a DPT vaccine, and somebody can report that. It doesn't mean that it's causally associated. In contrast, an adverse events of special interest are pre-specified medically significant events that you have reason to think could potentially be causally associated with vaccines. And you may identify these for things you want to monitor because there's already a proven association with immunization, such as anaphylaxis, because there's a proven association with a known vaccine platform or adjuvant, theoretically based upon what you know about the disease that you've developed the vaccine for. If you're using a live attenuated vaccine based upon viral replication, something you might have learned in animal models, or again, looking in special populations like pregnant women. So there is a spectrum of safety monitoring. And again, Tom is going to show you in the examples in the US of how this actually works. But at the one end of the spectrum is passive surveillance, where anybody, healthcare worker, or somebody from the general public, can report any adverse event following immunization, whether or not it's related. We know that it may or may not be associated with vaccines, and underreporting is common, but it is a way of identifying, in particular, unusual events that may be occurring. The other end of the spectrum is active surveillance, where you're doing a systematic search or scan for defined adverse events in targeted populations such as clinics, hospitals, and communities. All countries should, at a very minimum, be able to do and be conducting passive surveillance, and then ideally reporting their data to WHO's global database. And this was as of last August. You can see that they had already had over 1.7 million individual case reports reported in the system. And not surprisingly, the most common events were those that were consistent with the clinical trials: headache, pyrexia, fever, fatigue, chills, myalgias, et cetera. So I'm going to be focusing on what we've learned about the WHO emergency use listed vaccines. As of now, these are the 7 emergency use listed vaccines. The 2 mRNA vaccines, Pfizer-BioNTech, known now by many as Comirnaty, the Moderna vaccine, also known as Spikevax, the 2 adenovirus vector vaccines. Note that I have a 3A and B. That's because the AstraZeneca Oxford, also known as Vaxzevria, and the Serum Institute of India's Covishield are the same formulation, but different manufacturers. And then fourth is the Janssen or Johnson & Johnson or Janssen or J&J, depending where you live. All of those pronunciations are used by the company. Because this is used in the US, Tom will be showing a lot of data on this. So I'm going to skip through some of the Janssen, and you'll see what I mean when we get there. Then there are 3 inactivated vaccines, 2 from China, Sinopharm and Sinovac, and the most recent addition to the EUL listing in November was Covaxin from India. Next week, SAGE is meeting to discuss Novavax, which makes me think, I don't have any inside information, but it makes me think that WHO must be getting close to doing an EUL listing for Novavax. So how does WHO headquarters do their post-marketing surveillance? Again, I'm not from WHO, so I'm going to be giving you my perspective from the GAC side. They're getting information from a variety of informal, formal sources. They're also meeting regularly with their WHO regions. They maintain a watch list of things that they want to keep an eye on or they have concerns about. And any that are raised to a certain level, they bring to this GAC's COVID-19 subcommittee that was stood up about a year ago in order to be able to provide independent advice to WHO. So I've got the next 2 slides are examples of the watch list. This is from back in August, but it hasn't fundamentally changed, so I didn't ask them to update it. You don't need to be able to read all this. I'm going to walk you through the most salient points. So they have the topic of interest, such as Guillain-Barré, which vaccines are related, and then the status of the signal, whether it's been confirmed or ruled out, whether the GAC subcommittee reviewed it. If you see something in blue, it means that the GAC subcommittee issued a statement that was put on the WHO website about the status of the signal. So what I want to show you is in pink, there were 3 confirmed signals. Anaphylaxis, not a surprise. You would expect that with any vaccine or therapeutic. And Tom will show you more data, but for instance, the mRNA vaccines, it's running at about 5 per million for anaphylaxis. Thrombosis with thrombocytopenia syndrome after the adenovirus vector vaccines, and we'll talk more about that in a minute. Myocarditis, pericarditis after the mRNA vaccines, and again, we'll talk about that more in a minute. The next level is product warnings, where it's the regulatory agencies didn't actually confirm the signal yet, but there is enough concern that they wanted to put it on the label. That includes Guillain-Barré for the adenovirus vector vaccines and capillary leak syndrome for the adenovirus vector vaccines. There are a number of other things that have been raised but have not been confirmed yet. And then there are some that are in green, what you would expect based upon the clinical trials, such as flu-like symptoms after the AstraZeneca, delayed local reactions such as COVID arm after Moderna, deaths in the frail elderly in Norway after the Pfizer Comirnaty. And I want to tell you that, in my opinion, I think it's still important, even if you think that this is what you would expect, to investigate these events, both because you want to make sure nothing more serious is going on, and because they can have a potential detrimental effect on your immunization program. For instance, with the flu-like symptoms in Sweden, several of their health districts had actually paused vaccination when about 25% of their nurses were calling in sick. So now I'm going to focus on the most serious safety findings that have been identified. But first, I want to mention that all of these EUL listed vaccines continue to show a positive benefit risk profile. Their common adverse events are consistent with what has been seen in the clinical trials. Local reactions such as pain and redness, systemic reactions such as fevers, myalgias, headache, nausea, fatigue, etc. We already talked about anaphylaxis, so I'm going to start with adenovirus vector vaccines with thrombosis with thrombocytopenia syndrome, or TTS, and Guillain-Barré. So for TTS, back in March, Denmark paused vaccination after reports of severe blood clots. Several other regulatory agencies followed suit, and this was rapidly investigated in March amongst the early adopters of the AstraZeneca vaccine, including the European Medicines Agency, the UK, India, as well as a review of the global database. At the time, the GAC subcommittee conclusions were that the vaccine continued to have a positive benefit risk profile. They didn't identify any overall increase in clotting conditions such as deep vein thromboses, but they did note these very rare and unique thromboembolic or clotting events in combination with thrombocytopenia or low platelets, such as in rare vessels, such as the cerebral veins and cerebral venous sinus thrombosis. And Tom will show you a nice picture of what that actually looks like. So over the next month, there was intensive continued investigation, and what they determined after about a month in mid-April was that these appeared to be very rare events in the order of about 1 to 2 per 100,000, but it did seem plausible that it was associated with vaccination. The overall the benefit risk profile remained positive, but a biological mechanism was already being investigated and hypothesized related to platelet factor 4 antibodies, which is seen with heparin-induced thrombocytopenia. This led to the recommendation early recommendation to avoid the use of heparin and to use other types of anticoagulants. At this time, a platform-specific mechanism related to adenovirus vector vaccines could not be certain, but it couldn't be excluded. And sure enough, shortly thereafter, the US that had just started using the J&J or Janssen vaccine identified what appeared to be TTS in mid-April after use of the Janssen vaccine. Because of the collaboration and cooperation amongst the various regulatory and public health authorities, it was rapidly determined that this was the same clinical syndrome that was being seen in particular in the European Union with the AstraZeneca. And both the European Medicines Agency and the US were able to rapidly confirm an overall positive risk benefit and emphasize the importance of early recognition and appropriate management. And again, Tom will be showing you more data on that from the US. The WHO and many other countries also rapidly developed clinical guidelines to how to manage this to reduce severe morbidity and mortality. They're in the process of updating these based upon new information that has been developed, that has come to light over the last 6 months. So stay tuned. But what have we learned about TTS since then? This is data from the UK's passive surveillance system called the Yellow Card as of the end of November. And what you can see is after the first dose, their reporting rates are running at about 1.5 per 100,000 after the first dose. And you can see that I think one of the largest users of the AstraZeneca with over 24 million first doses and over 24 million second doses. So you can see that this rate is still around the order of what we were talking about back in April, about 1.5 per 100,000. But you can see it's markedly lower after the second dose, 0.2 per 100,000 after the second dose. So that's about 7 or 8-fold lower. They also have found a higher incidence in those under the age of 50 after the first dose, and their overall case fatality is running at about 17%. Most of these are after the first dose, only 6 were after the second dose. As another example, this is from Australia over the summer. Their reporting rates are in the same ballpark, between 1.8 and 2.9 per 100,000, also seeing a higher rate in younger people and more severe in younger women. I'm going to skip over the Janssen data since Tom will show it to you, but to say that they are seeing a similar picture in the US with the Janssen vaccine. So an example of an adverse event of special interest that is showing itself in the same platform of vaccines. There have been a couple special studies that have been, there are many studies, but a couple special studies I wanted to point out that have been published. One from Oxford showing that while the overall mortality is low, similar to what was shown in the passive surveillance system in the UK, here it was 22%. The mortality was much higher at 73% when people had significantly low platelets under 30,000 and evidence of intracranial hemorrhage or a head bleed. So again, the importance of early recognition and management. The European Medicines Agencies wanted to look and see whether they identified risk factors that are normally associated with clotting, such as birth control pills use, and they did not find that these were associated in their population. In the Oxford study, they also looked to see which vessels were affected. And as we said, cerebral veins was the first thing that really stood out because it's an unusual vessel. But they noted that it can be a large variety of vessels. So it's not just, you're really looking for blood clots. And Tom will go through the case definition in more detail. So what else have we learned? And I'm not going to spend a lot of detail on these, but I did want to mention that there have been a couple studies that have come out very recently looking at the biological mechanism. And in this study, they proposed that it's actually a structural part of the adenovirus that seems to be binding with antibodies to platelet factor 4. And in contrast to originally what people were wondering, could it be cross-reactivity with the SARS-CoV spike protein? And the answer in this study was no. So, which would explain why we're not seeing this happen with the mRNA or the inactivated vaccines. So moving on to Guillain-Barré, this is again data from the UK's passive surveillance system, where you can see that they've had over 475 cases of Guillain-Barré syndrome or Miller-Fisher variant, in contrast to very low rates with Pfizer and Moderna, which I would assume is likely their background rates. A similar story is seen in the US. And again, I'm going to skip that with Janssen because Tom will go through it with you in detail. But just to say that the two vaccines seem to be mirroring each other for both of these, both TTS and Guillain-Barré. Moving on to the mRNA vaccines, I wanted to focus on myocarditis, pericarditis. Israel was the first country to report a possible link between the Pfizer second shot and heart problems in men under the age of 50. Then it was identified in many other countries. And I'm going to give you a couple of examples of what we've learned. Again, Tom will be going through a lot more data in detail on the US's data. This is from Canada, a paper that was just uploaded a couple of days ago. Of note, this is a preprint, so it has not yet been peer reviewed. So I don't want to sort of completely hang our hats on this data, but I do think it's important data. And what they're finding, which is consistent with what most other countries are reporting, is that most of these cases are occurring after the second dose. Their median time is 3 days after the second dose, and most are occurring within 7 days. Most of them are occurring in men, and it's occurring in young men. Their median age was 24. So their highest reporting rate was in 18 to 24-year-old men after the second dose. And in particular, they found it was after Moderna. They found a 5-fold higher rate after Moderna than after Pfizer. Still a very rare event. If you look, it's about 300 per million. So if you figure that's 30 per 100,000 or 3 per 10,000 versus 59 per million, which would be about 6 per 100,000 or 0.6 per 10,000. So still a very rare event for both of these vaccines, but occurring at least in Canada with a higher rate after Moderna than Pfizer. Other countries are also following not as high as five-fold, but a 2 to 3-fold in some countries. Other places are not finding this differential rate. So I think people are still trying to determine, different places are weighing more heavily about whether there's a differential rate. And you can hear from Tom what his opinion is on that. Overall, the reporting rates for Canada seem to be higher when the interdose interval was shorter for both vaccines. Canada is one of the few countries where this was actually able to be looked at because they had a fair number of people who both got both doses within 30 days or had them after 8 weeks. So they found a longer interval was associated with a lower rate of myocarditis. But they did find that regardless of what first dose was used, they did find that there was a slightly higher rate with Moderna than Pfizer. It wasn't a huge difference, but it was slightly higher. Also, with just a few days ago, the European Medicines Agency, which is a regulatory authority for those who are not familiar with it, posted their updates to myocarditis, pericarditis. And what they report is that both of these conditions are overall very rare, which is a definition of about up to 1 per 10,000 vaccinated people. They reviewed in this recent update data from 2 studies, from French and from the Nordic. The Nordic study has not yet been published. I think people are actively waiting for it. It's been in the news over the last month or two. They again confirmed extra cases in younger men following the second dose, and they looked at within about 14 days. You can't directly compare these two studies because their populations and their time intervals are a little bit different. You can see in the French study, it's 12 to 29-year-olds. In the Nordic study, it's 16 to 24-year-olds. The French study, it was 7 days after the second dose. In the Nordic study, it was within 28 days. But you can get a sense of it. So in the French study, this is after Pfizer, they had 0.26 cases per 10,000. So that would be what, about 2.6 per 100,000 or 26 per million. For the Nordic, it was 0.57 per 10,000. So again, that would be about 5.7 per 100,000 or 57 per million. I give those different rates because different people process these differently. For Moderna, it was slightly higher, but still low, 1.3 per 10,000, so 13 per 100,000 or 130 per million, and 1.9 per 10,000 out of the Nordic study, or 19 per 100,000. They felt that the benefits of all these authorized vaccines continue to outweigh the risks. And as I said, they felt that for both of the vaccines, it was still rare. This was coming from the US's Advisory Committee on Immunization Practices, a colleague that consulted with Tom's group, gave a background on myocarditis for the Advisory Committee, or ACIP, which I thought would be helpful for you to see that in a pre-COVID era, the highest rate of myocarditis tends to occur in older adolescents and young adults. If you look at older adolescents in the left-hand column, I don't know if you can see my arrow or not, so just in case. The young adults, you can see that the rate starts to go down at 20, 25, 30, 40. And then in this study, in this slide, he also presented at ACIP. He was showing us the differential effect of myocarditis in different syndromes. And the blue is what we're looking at on the y-axis is the ejection fraction, which is the amount that the heart is pumping. And 100% means that 100% of people had normal ejection fraction. So what you see with the blue, which is classic non-COVID, non-vaccine myocarditis, what you see is about 2/3 of people return to a normal ejection fraction after myocarditis. In red, what you see is over 90% of people returning to normal after having multisystem inflammatory syndrome in children. And what you see in green is the vaccine-related myocarditis. And what you see is that the percentage of people who return to normal ejection fraction is much higher, and it is occurring much more quickly, suggesting that it's a milder, more self-limited myocarditis. And Tom will go through this in more detail. The other thing is on the safety, which is what Tom and I are focusing on today. We're focused on the risk side of a benefit risk assessment. But as a reminder, we always need to think about benefit risks. And this was a paper published from Israel over the summer, showing that their rate of myocarditis after vaccination was about 3 per 100,000 person years, but their rate after COVID disease was about 11 per 100,000 person years. So now moving on to the inactivated vaccines. To date, the data for all three of these is reassuring, and I'm going to give you some examples, but I will say that the surveillance data is just not as mature as what for the vaccines I've just shown you. I'm going to start with Sinovac, and as an example, give you data from Brazil. You can ignore the top few rows, which are COVID related. And what I just want you to be able to see is on the left for serious adverse events and on the right for deaths, that you just don't see a differential. You don't see any kind of clustering of any particular illness. And then they had received reports of deaths in the elderly in long-term care institutions. So they did an investigation. And what they found was that these deaths were not causally associated with vaccination, and they had no increase in the baseline death rate. Again, showing the importance when you get these reports of actually doing an investigation. And in this case, it was very reassuring. Moving on to Sinopharm, this is data out of Morocco where they've used over almost 13 million doses as of July and had over almost 5,700 adverse events following immunization. But if you look, only 0.6% of them were considered serious, or 35 of them. And the most frequent was not surprisingly anaphylaxis, but all 35 of these had a favorable outcome. So that was very reassuring. COVAXIN, which is the newest player, this WHO Strategic Advisory Group of Experts, or SAGE, that puts out the immunization policy recommendations for WHO, asked the GAC subcommittee to review the COVAXIN data prior to their making their recommendations for SAGE. And so the GAC subcommittee in particular reviewed data from India, where they had given over 77 million doses. And I don't have the approval to provide you with the actual data. So what I'm going to do is provide you with our summary from it, which were that there were no serious safety concerns identified, and the data were very reassuring. I will note that, like for much passive surveillance data, that the data was limited by underreporting and a lack of reporting after 7 days. However, I will say and I give kudos to India, that they were in the process of setting up a number of active surveillance studies, pregnancy surveillance, pregnancy studies, and upgrading their reporting tools so that they could get better data. And then finally, I do want to mention special populations, in particular pregnant women. Tom is going to show you the specific data, since much of it is from the US. But I did want to mention that we're paying particular attention to this because the pregnant women were not specifically included in pre-authorization trials, although there were no safety signals identified in the animal developmental and reproductive toxicity, or DART studies, which was reassuring. Also, the J&J had a previous adenovirus vector vaccine, such as the Ebola vaccine that is licensed in the European Union. And they did study pregnant women and identified no safety signals. Tom will show you the specific data, so I won't, but I will say that the data for all these vaccines to date is very reassuring. And recently, WHO SAGE changed their recommendation from considering vaccination of pregnant women to a full-on recommendation to vaccinate pregnant women because of the potential severity of the disease in pregnant women, the risk to the unborn child, and the safety profile of these vaccines. So again, all of these vaccines, the safety profiles for the common adverse events are consistent with the clinical trials. Our surveillance systems are able to detect rare events such as anaphylaxis, TTS after the adenovirus vector vaccines with a rate of about 1 to 2 per 100,000, possibly in younger populations, in particular women, Guillain-Barré syndrome after the adenovirus vector vaccines, and myocarditis, pericarditis after the mRNA vaccines, again in younger males within a week after the second dose. Most of these seem to be self-limited, though longer-term follow-up is needed and is underway, and you'll hear about that from Tom. There are a number of challenges, which I think you're all probably very familiar with. The quality of the surveillance in different countries and settings is variable. There are multiple vaccines being used in most countries, and oftentimes simultaneously. They're being given as quickly as they can, which means that background events, it can be challenging to determine if these are background rates of illness or death, or whether they're vaccine related. There are also a number of questions that continue to come up. Many of these have been answered or are being answered, such as the safety of homologous and heterologous vaccines. And I will say that the data is very reassuring to date. Vaccines with different variants that are being developed in previously infected individuals, which again, data is reassuring to date. Safety of different timing of vaccines, such as what we were seeing in the Canadian data. Difference in different populations, such as the elderly, pregnant people, and long-term follow-up of myocarditis. But overall, the safety profile of these vaccines is reassuring. WHO is continuing to monitor the safety of all these vaccines, and the GAC subcommittee is continuing to meet regularly and advise WHO. And I do want to close by saying that communications needs to be open, transparent, and data-driven. Most of this data and many of these slides come from colleagues around the world, for which I am very grateful. And I want to thank you for your attention and hand it back to Melinda. Is it going back to you, or is it going to Tom directly?
Well, I think we'll move straight on to Tom's presentation. So thanks so much. And Tom, do you want to get your slides up?
Yes. I'm trying to figure out how to unshare, stop share. Okay.
And is the big screen up?
Yes, you're in presentation mode now.
Okay. Thanks. Hi, I'm Tom Shimabukuro. I'm the acting director of the Immunization Safety Office at CDC, and that's the office that does post-authorization or post-licensure safety monitoring. And it's a pleasure to speak on the ADVAC alumni webinar. I appreciate the opportunity. Here's a disclaimer. I just want to mention that much of this information and additional information is taken from presentations that have been given at meetings of the Advisory Committee on Immunization Practices and also on FDA's VRPAC meetings. And just mention that we do have a CDC COVID-19 publications website, which includes pretty much all the publications that CDC has put out on COVID-19 vaccine safety since the beginning. We also post our protocols and our standard operating procedures for our surveillance and our research activities on the website there. So I'll cover a bit of background, and I think Rita covered most of it, so I may move through it fairly quickly, and then describe the CDC surveillance systems and processes for monitoring COVID-19 vaccine safety. I will mention that this is from the US perspective, but it's largely focused on CDC. FDA is our surveillance partner, and there are other groups that also do vaccine safety surveillance in the United States, the Department of Defense, the Departments of Veterans Affairs, and the Indian Health Service. But this presentation is going to be primarily focused on CDC systems and CDC results. And then I will describe in more detail some selected adverse events of special interest and pre-specified outcomes for COVID-19 vaccine safety monitoring. And this is really going to be doing a deeper dive into adverse events for which we believe we have a sufficient body of evidence that we have been able to establish a causal relationship, or we have evidence that is highly suggestive of a causal relationship. I'm also going to touch on pregnancy outcomes just in general. So again, Rita showed you this vaccine life cycle infographic, just to mention where CDC's role is really in that lavender chevron you see there. After the basic research and the clinical trials and the FDA review and the authorization or licensure, shortly thereafter, there's an ACIP review and a recommendation by ACIP. And then we move into phase 4 or post-licensure, post-authorization, also called post-marketing. And that's where the Immunization Safety Office at CDC begins to do its work. Rita also covered some of these issues. One thing that I want to emphasize is that one of the main reasons that we do monitoring in the post-authorization period is that the clinical trials don't always include some of these special populations, in particular pregnant women and people with certain pre-existing medical conditions. But we know that people with pre-existing medical conditions are actually prioritized for vaccines. And we know that that pregnant women are getting vaccine. In fact, CDC has recommended that. So really, the post-authorization or post-licensure monitoring is the only way to get safety data on these individuals. But also expectations are especially high that public health and regulatory agencies will intensively monitor the safety of vaccines administered under emergency use authorization during a large-scale national vaccination program. So this shows the CDC monitoring systems or infrastructures or programs, the main ones. And we are monitoring these vaccines under the most intense safety monitoring effort in US history. These systems are complementary. It includes the vSafe system, the Vaccine Adverse Event Reporting System, the Vaccine Safety Data Link, and the Clinical Immunization Safety Assessment Project. And I'll describe these in sequence here in the following slides. So the first system that I'll cover is vSafe, which is a new system that was established just for COVID-19 vaccine safety monitoring. This system actually didn't exist prior to the pandemic. It is a smartphone-based system that uses text messaging and secure web-based surveys to check in on individuals in the post-vaccination period to assess their post-vaccination experience. The history behind vSafe is that when we were in the planning stages in the summer of and fall of 2020, we realized that VAERS is our frontline system, a very responsive system, an established system that has been in place for over 3 decades and has really proven its utility. However, VAERS is a spontaneous reporting system, and it relies on individuals to send reports into VAERS. We also have our large link database systems like the Vaccine Safety Data Link, and some FDA systems like the monitoring in the Medicare data, and also the FDA BEST system. And these are population-based systems. However, it takes substantial time to get vaccinations into the system, and also you need follow-up time to assess the outcomes. So while VAERS is very responsive, it's a passive surveillance system. While these large link database systems are high quality, it takes time to get meaningful data, and there are issues with data lags. We felt like we needed to establish a more active system where we could get almost immediate feedback as soon as these vaccines were administered. So we established vSafe, which is a CDC smartphone-based monitoring program for COVID-19 vaccine safety in the United States. It uses text messaging and web surveys to check in on recipients after vaccination. It is a voluntary self-enrollment program that requires the individual, or in the case of minors, the parent or caregiver to have a cell phone. I guess those are some of the limitations of vSafe. It's voluntary, it's self-enrollment, and you have to have a smartphone to be able to participate in the program. Participants can register anytime. So after the first or the second or the third dose. However, you can't go back in time. For example, if you register after your booster dose, you can't go back in time and report on first and second dose. Those surveys for those surveys are basically time limited, and I'll explain that a bit in the next slide. Parents and guardians can register on behalf of children. vSafe solicits participants' reports on how they feel after COVID-19 vaccination, which mainly includes local questions on local injection site reactions, systemic reactions, and health impacts. And health impacts are defined as inability to perform normal daily activities, missing school or work, or receiving care. So this just shows sort of the information flow, or the flow of vSafe. Again, individuals voluntarily register. We like them to register when they get vaccinated. However, they can come in as a lateral entry really at any time. And individuals get text messages daily during the first week following vaccination with embedded links to secure surveys. So they get those daily for the first week, and then weekly through 6 weeks, and then at 3, 6, and 12 months. So these, again, as I mentioned, these surveys are time limited. So if you don't respond to your first daily survey by midnight on the first day, you have to move on to the next survey. You can't go back in time and complete a previous survey. If an individual reports a clinically important health impact event, and we define that as receiving medical care, that person's information is transmitted to a vSafe call center, and we'll reach out to that individual and we'll take a VAERS report if appropriate. We also have a pregnancy registry team that's conducting outreach to assess eligibility for the registry and obtain consent for enrollment and follow-up, and I'll discuss that in a little bit more detail. So this is a table from a publication on vSafe data, and you can see here some of the local and systemic symptoms that we ask about, and these are asked about during the first week of vaccination. Again, we also ask about health impact events, which are really more quality of life issues. Those aren't reported here. This is just showing reactogenicity, and I will mention I think vSafe initially, when it was initially conceived, we thought it was going to provide substantial value added for basically stimulating reports to VAERS for more serious adverse events or clinically important adverse events, and I think it has done that. But I looking back on it, I think some of the real value of vSafe is it really was able to confirm the basic safety profile, the basic reactogenicity of the vaccines as they were being administered in real time. And I think some of the take homes from vSafe is that we learned that the mRNA vaccines are quite reactogenic. People experience substantial local and systemic reactogenicity. They also experience substantial health impact events with respect to not being able to do daily activities or missing work, and in the case of children, missing school. And that's consistent with what was observed in the clinical trials. What we also observed, and I think was consistent with the clinical trials and was, I think, consistent with anecdotal reports during the immunization program, is that there was more reactogenicity after Moderna compared to Pfizer, especially after dose 2. And we've seen this, basically it's consistent after dose 1, after dose 2. Now that we're monitoring boosters, also more reactogenicity after booster doses for Moderna compared to Pfizer. With respect to booster doses, and this has been reported out at ACIP, we're actually seeing for both Moderna and Pfizer less reactogenicity following the booster dose compared to the second dose, which is generally reassuring. So now I'm going to describe the vSafe pregnancy registry. So there are a series of questions in the basic vSafe surveys that assess pregnancy status, where you're either pregnant at the time of vaccination, and you knew it, or you found out you were pregnant later, and when you were vaccinated, or you were vaccinated in the periconception period. When we get information on these individuals that answer in the affirmative to these, we reach out, we screen them for eligibility, and if we determine they're eligible, we'll consent them for registry enrollment, and we will follow up on them at all stages in pregnancy and early in infancy. Sometimes that's a retrospective follow-up because we don't always get to them during the trimester that they're in. And to date, we've enrolled about 7,200 individuals, pregnant women, in the vSafe pregnancy registry. So now I'm going to move on to the vaccine adverse event reporting system, which I think most of you are familiar with. VAERS is the US's early warning system for vaccine safety. It's a spontaneous reporting system or a passive surveillance system that's co-managed by CDC and FDA. There are basically two kinds of reports to VAERS. There are public reports and there are manufacturer reports. The manufacturers report through a separate process using the E2BR3 data elements, and that's managed by FDA. The public reporting takes place largely through a fully automated process that's online. And what I've shown you here is the landing page for VAERS. If you go to HHS or vaers.hhs.gov, it will take you to this landing page, and you see up there on the top ribbon, if you click on report and adverse events, you will eventually get to a screen that looks like this. And this is the HTML VAERS reporting form, where you basically just go add in these, you fill in these blanks and then you submit. You don't have to, if you don't have information, you can still advance. We don't require any information on this. We'll take whatever the person can provide. There is also a fillable PDF form if a person can't use this online reporting form. You do get timed out of this system for security reasons. So a person can do the fillable PDF and upload that. That's another way of reporting. So VAERS accepts all reports from everyone, regardless of the plausibility of the vaccine causing the event or the clinical seriousness of the event. So we accept reports from patients, parents, caregivers, healthcare providers, and others. We encourage reporting, even if the reporter isn't sure if the vaccine caused the adverse event. The key strengths are we can rapidly detect potential safety problems. We can detect rare adverse events. The key limitations are consistent with spontaneous reporting in general. It's inconsistent quality and completeness of information, reporting biases, and a general inability to determine causality. This is a page from the VAERS standard operating procedures for COVID-19 vaccine. It includes some VAERS adverse events of special interest. And these adverse events of special interest you see here on the left are reports that we follow up on. So we'll contact the reporter or the healthcare facility to get medical records. We'll review those records. We'll apply a case definition that either meets the definition or doesn't meet the definition, or sometimes insufficient evidence. If there are questions about the diagnosis in certain cases, we'll adjudicate these by committee. Just want to mention that TTS is not on here because there really was no such thing as TTS prior to the pandemic, at least that we knew of. That's something that's been added. And so, we have the ability basically to adjust and add adverse events as need be. VAERS analysis includes descriptive analysis, empirical Bayesian data mining, which is done by FDA to detect disproportionality. Also observe versus expected, or observed reports compared to background, and individual report reviews. So moving on to the vaccine safety data link. This is our large link database system we use for active surveillance and research. And it's a collaboration between CDC and 9 participating integrated healthcare organizations you see here, and has data on over 12 million persons per year. So VSD is an electronic health record based system. It includes complete or near complete information on a patient population, which has immunization records, outpatient ED, hospital visits, procedure codes, birth and death certificate information, and enrollment demographics, all linked by unique study IDs. As an EHR-based system, we also have rapid access to charts, so we can go in, pull charts, review charts, confirm diagnoses, confirm incident or prevalent cases. We can usually do these quick chart reviews on a very short turnaround, usually a matter of a few days. Complete chart reviews may take a little bit longer, maybe a week or so, but generally speaking, if we do need to go in and assess a signal or go in and review individual case reports, we can do that rapidly in VSD. In VSD, we conduct rapid cycle analysis, or RCA. That's near real-time sequential monitoring. As data become available, we basically collect and refresh the data on a weekly basis and run a standard analysis. We monitor a limited set of pre-specified vaccine safety outcomes. So unlike VAERS, which monitors basically everything that comes in VSD, we have pre-specified these outcomes. We have a reason to look at them. So it's a bit more of hypothesis testing than data mining. It's a public health surveillance activity, which is not the same as an epi study. It's designed to detect statistically significant associations and statistical signals, which do not necessarily indicate a safety problem. Statistical signals detected through RCA require further evaluation. I mentioned one of the key things we do to evaluate signals is to go in, pull those charts, and quickly review those cases, determine if the diagnosis is valid, and also, also determine the onset with respect to vaccination, which is not always adequately documented in the medical record, which I'm sure some of you have experienced that. So the aims of COVID-19 RCA were to monitor the safety of these vaccines weekly using the pre-specified outcomes of interest, which I'll show you in a second, and to describe the uptake of COVID-19 vaccines over time. These are the VSD COVID-19 vaccine pre-specified surveillance outcomes. There's just over 20 of them. You see the outcomes there in the first column, the settings in which we're monitoring these outcomes. In the second column, that's either emergency department, inpatient, and in some cases, outpatient. The risk windows, and then some exclusion criteria. So some of these outcomes are, as you know, are associated with COVID-19 disease. So if an individual has COVID-19 in the prior X days you see there, we actually exclude the case. The ones that are in black font are ones that we're doing sequential monitoring on, so statistical testing sequentially. The ones that are in the blue font are ones that we are monitoring descriptively. And I'll use anaphylaxis as an example. So we know that anaphylaxis is associated with vaccines and medicines and other medical products. It's basically an exposure-driven outcome. And so we decided that we weren't going to do any comparisons there. We were basically on a weekly basis, we are going to update the rates and just descriptively monitor that, because we didn't really see much value in doing sequential monitoring. Again, because we know anaphylaxis is associated with vaccination. We know it's an exposure-driven outcome. This is a slide showing the main analysis, which you call the vaccinated concurrent comparator. I'm not going to get into the details of this. I might end up confusing myself, but basically the main analysis for VSD rapid cycle analysis for COVID-19 is comparing vaccinated individuals to other vaccinated individuals, looking at incidence in a risk interval compared to incidence in a control interval. It's not a self-controlled risk interval analysis where an individual serves as their own control. We really couldn't do that because we have these overlapping risk windows with these vaccines given at second dose at 21 and 28 days. So we're basically comparing incidence in a risk interval in individuals compared to incidence in a comparison interval in other vaccinated individuals matched on certain characteristics. So lastly, I'll talk about our clinical immunization safety assessment project. Was a collaboration between CDC and 7 participating medical research centers with vaccine safety experts. Our CISA project does clinical consult services and clinical research. CISA core activities for COVID have included clinical case reviews and clinical consults on complex cases of vaccine adverse events. We've actually established some work groups within CISA, for example, the hematology work group, where we take some of these complex TTS cases and we have a grand round style presentation of the case and get feedback from our consultants on whether this meets case definition or not. Also technical consultation on clinical guidance and clinical considerations, contributions to enhance surveillance for adverse events. CISA has also contributed to developing case definitions for TTS and for myocarditis, and then clinical research. So also mention that we have what's called the COVID-19 Vaccine Safety Technical Work Group, also called VAST. This is a work group in ACIP. It's actually a subgroup of the COVID-19 work group, which is focused just on safety. And this serves as a central hub for the technical subject matter expertise from the federal agencies that are conducting the surveillance. Bi-weekly includes bi-weekly or weekly review of data on adverse events. It's shared learning experience for all members. On these meetings of the VAST work group, we review, evaluate, and interpret post-authorization safety data. The VAST advises the federal agencies on analyses, interpretation, and data presentation. Following the data presentation part of the meeting, there is a closed session where the VAST members meet together and discuss findings. And we also provide updates to the COVID-19 vaccines work group and to the ACIP on COVID-19 vaccine safety. So now I'm going to cover some selected adverse events of special interest and pre-specified outcomes. And again, I've chosen these adverse events because we have substantial evidence of a causal association. And then I'm also just going to talk briefly about pregnancy and infant outcomes. So the first outcome I'll talk about is anaphylaxis. And I'm talking about mRNA COVID-19 vaccination. I mean, there is anaphylaxis after Janssen, but the experience after mRNA vaccination, I think, is instructive. So in the week after the rollout of the US vaccination program, at CDC, we began to get reports and direct outreach from healthcare providers on what seemed like an unusually high number of allergic reactions and anaphylactic reactions after mRNA vaccination. It started with the Pfizer vaccine because that became available a week earlier than the Moderna vaccine, but really after both mRNA vaccines. And it was concerning to us because, again, it seemed like a high number of reports relative to what we might expect to see from other vaccines, particularly that are given out in large quantities in a short period of time. And I'm thinking mainly of flu vaccine. So these suspected reports in the early stages of the program were detected through early screening. So basically pre-screening VAERS reports prior to the formal processing and med-recoding. Our VAERS contractor would look at them, and anything suspicious of allergic reaction or anaphylaxis the report was sent directly to us prior to it even getting a formal processing and review and med-recoding. We also detected these through the normal process of a med-recode search after processing and coding in the VAERS database. Suspected anaphylaxis reports were assessed by physicians at CDC, who did medical record review and conducted additional follow-up to include getting additional records or talking to the healthcare provider. And then we used the Brighton criteria to classify them. And CDC and FDA met daily, actually, to further adjudicate cases if necessary. So this is a report that came out in the data from this was from December, and it came out in early January, and it was looking at the reporting rates or the rate of anaphylaxis after the Pfizer vaccine. And at that time, we had estimated a rate of 11.1 cases per million doses, which is high, at least compared to what we expect for other vaccines. I'll show you some data where I think there may have been some biases in the data, possibly some stimulated reporting, possibly maybe some what I would call overdiagnosis. Many of these early recipients of vaccine were healthcare providers, and they got vaccinated right down the hall from the emergency department. So probably any adverse event suspicious of allergic reaction or anaphylaxis was treated aggressively. Plus, at the time, this was early on in the program, and I think the states may have been in the process of learning the best way to report doses administered, so denominator data to us. So we may have had undercounted the denominator. But anyways, more to come on this initial rate for Pfizer. This is a similar report from the MMWR looking at Moderna. You can see at 2.5 cases per million doses, so substantially lower than for Pfizer. And then several weeks later, I think after things maybe had settled down, we did basically a refreshed analysis, and this is from a JAMA article, looking at the reporting rates for both Pfizer, BioNTech, and Moderna. And at that time, which basically had data through mid-January, where the data had settled a little bit, we had a reporting rate of 4.7 per million doses administered for Pfizer and 2.5 for Moderna. We are also monitoring in our vaccine safety data link a population-based system, which is probably a better database to capture information on anaphylaxis. And the initial estimates for the anaphylaxis rates were 4.8 for Pfizer and 5.1 for Moderna. So basically pretty similar, and around 5 per million doses administered. So just to put that rate in perspective, and this is from a paper by a couple folks in the Immunization Safety Office, looking at rates of anaphylaxis after a lot of vaccines here, and I think the reason we were initially concerned with anaphylaxis was because our baseline is largely anchored with flu vaccine, which is about 1 to 2 cases per million doses administered, and we were seeing 5 or more. But when you look at the rates of a lot of other vaccines, you can see that 5 per million really is within the range of what we might expect for other routinely administered vaccines. So I think our conclusion now is, although the anaphylaxis rate may be higher than for inactivated influenza vaccine, it's within the range of what we might expect for a lot of other vaccines that are routinely given in the US. So moving on to TTS. So I think Rita mentioned that the initial signal for this came out of Europe, where there were these unusual cases of blood clots with low platelets following the AstraZeneca vaccine. Just to remind you of the cerebral venous sinus anatomy, if you're somewhat removed from gross anatomy in med school, this is the venous circulation of the brain. You have these large sinuses here. And from my understanding is not only are these sinuses large, there's a lot of redundancy in the drainage. And so when you become symptomatic, at the point you become symptomatic, you're already have a serious medical condition, because again, it takes a fairly large clot to have a substantial impact on the cerebral venous drainage of the brain. So this is the Janssen timeline, and EUA for Janssen was given in February 27th, and then the recommendation by ACIP on the 28th. Then on March 2nd, vaccination started. Then on March 19th through the 12th, CDC and FDA detected 6 cases of CVST with thrombocytopenia. On the 13th, CDC and FDA recommended a pause, and a Health Alert Network alert was issued, and the investigation continued. That's a typo there. April 13th, it should be sometime in the 20th. I'll have to adjust that, but it was maybe a week after the pause. ACIP reviewed the data and reaffirmed the recommendation for Janssen vaccine, and surveillance continued. So this is a screenshot of the CDC Health Alert Network. And I think the reason that CDC and FDA felt this pause was necessary is number one, this was a serious health condition. There was biological plausibility for this outcome based on the data in AstraZeneca. It was very unusual, and probably, and we needed time to assess the significance, but probably importantly, the normal treatment for thromboses, which is to give heparin, in this case, may have caused patient harm. So we felt like we needed time to get the information out to healthcare providers. Number one, to be alert if a patient comes in with these signs and symptoms of potential CVST, work them up, keep the differential in mind, and do not treat with heparin until you rule out TTS, basically. treat with other non-heparin anticoagulants until you establish the diagnosis. So these are the CDC working case definitions for TTS. We basically have tier 1 and tier 2. Tier 1 is a thrombosis in an unusual location like the cerebral venous sinuses or the portal vein or splanchnic veins. And a platelet count of less than 150. A positive heparin PF4 ELISA hit antibody test is supportive, but was not required for a TTS case. For a tier 2 TTS case, thrombosis in a common location only was sufficient if there was a platelet count less than 150 and the person had a positive heparin PF4 ELISA hit antibody test. So we included this requirement because we wanted to increase the specificity in these tier 2 cases. So this is pretty dated information. It's from May. It shows reporting rates after Janssen by sex and age group, but I will say we do have more current data that I'm not at liberty to share, but the basic epidemiology has not changed. So the take home from this comparison here, looking at males to females, is that if you look at really at all age groups, the reporting rate per million doses administered is higher in females compared to males. And then if you look at this, what I've highlighted here, this is looking within females only. The reporting rates are highest in women aged 30 to 39 and 40 to 49, where it's 12.4 per million and 9.4 per million. Again, these values have changed somewhat, but the basic female preponderance and the highest risk in females in the ages 30 to 49 has been maintained really from the start. So these are some of the publications or notifications that have come out on TTS. And I'll mention that there is a preprint of a CDC and FDA case series of TTS following COVID-19 vaccination. I'll mention in that preprint, there are 2 cases after Moderna vaccination, but based on the doses administered, we likely think this is background spontaneous hit. And then on the bottom there, you see a screenshot from a CDC website where we report on adverse events. And I'll just read it there. It says a review of reports indicates the causal relationship between the Janssen vaccine and TTS, a rare serious adverse event that causes blood clots with low platelets, which has caused or directly contributed to 6 confirmed deaths. So now I'm going to move on to myocarditis after COVID-19 vaccination. As Rita mentioned, the initial signal was detected in Israel, which exclusively used the Pfizer vaccine. In the United States, our colleagues in the Department of Defense initially noticed what they thought was an unusually high number of cases in patients in their active duty service member population, really after both vaccines, both the Pfizer and the Moderna vaccination. At this point, evidence from multiple safety monitoring systems in multiple countries supports the finding of an increased risk of myocarditis and myopericarditis following mRNA COVID-19 vaccination. This risk is highest in adolescents and young adults, greater for males compared to females, and greater following dose 2 compared to dose 1. The onset clusters within a few days of vaccination, mostly within a week, and the cases have tended to be clinically mild. So here's some of the information globally that we have also reviewed and relied upon to inform our own analyses here in the United States. So now I'm going to show you some data from VAERS, and this data has been presented at a recent ACIP meeting. So this table looks at reporting rates per million doses administered of myocarditis or it's also myopericarditis among males after mRNA COVID-19 vaccines using a 7-day risk period. We've basically in VAERS started looking at the 7-day risk period because we think this is the most likely biologically plausible risk period. You see the age groups there on the left, and then in the first major column, Pfizer, followed by Moderna, dose 1 versus dose 2. Not calculating anything for Moderna in the 12 to 15 and 16 to 17-year-old ages because it's not authorized in the United States. Those peach colored cells are ones where the reporting rates exceed background incidence based on the background incidence from the literature. And you can see for males for Pfizer, we have background rates, we have reporting rates that exceed background in the ages 12 through 24 for dose 1, and then 12 through 39 for dose 2. And then for Moderna, we have reporting rates that exceed background for following dose 1 in ages 18 through 39, and following dose 2, ages 18 through 49. So this is the similar table, but for females. And I think what's notable here is, first of all, if you look back, if you look at these reporting rates, just sort of try to get a mental image of these reporting rates, especially after dose 2. They're substantially lower in general in females compared to males. Notably, there is no excess risk or nothing that exceeds background following dose 1 for either vaccine. And then for Pfizer, you see background rates that exceed reporting rates that exceed background incidence in the ages 12 to 24 for Pfizer, and then for Moderna, 18 through 29. I'll also just mention, if you look at these rates for Moderna versus Pfizer, you get a hint of a trend that the reporting rates are greater, where you can compare the reporting rates are greater for Moderna compared to Pfizer in males, slightly higher, and also slightly higher in females. The caveat here is this is spontaneous reporting, and these may have been given in different populations. But we'll get to some additional information from VSD. So we do follow up on these cases to assess outcomes, and this shows data on cases in persons less than 29. To date, or as of November, there are 1,640 total preliminary reports, 877 met case definition. Of those 877, 829, most were hospitalized. So in the United States, most of these cases get hospitalized, we think largely as a precaution. Of those 829, 789 have been discharged, and 607, or 77%, over 3/4, were known to have recovered from symptoms at the time of the report. So certainly one thing of concern is, have there been any deaths from vaccine-associated myocarditis? So we look at all death reports, but we closely look at these reports of deaths that were suspicious for myocarditis. As of late November, we had 9 reports with case reports of death with concern for myocarditis. And I'll just walk you through the branching diagram here. So one of those reports, there was inadequate information to assess. That person had myocarditis after vaccination, died, but the family refused an autopsy, so there wasn't sufficient information to assess other causes. So of the 8 remaining, the evaluation was ongoing in 2. And of the 6 where the evaluation was complete, 3 were not myocarditis. 3 were myocarditis, but there were other potential infectious causes identified. So the take home from this slide is there have been deaths with myocarditis following vaccination. However, we don't have conclusive evidence that would suggest that the vaccines caused or contributed to the deaths in any of these individuals so far. So moving on to the myocarditis analysis in VSD. So this is a table from a previous ACIP presentation looking at myocarditis and pericarditis in 12 to 17-year-olds, so Pfizer only, in the 0 to 7 and 0 to 21 day risk interval, looking at outcome events in vaccinated comparators. And so I think the take home here is regardless of the risk interval, regardless of the dose, you do see an elevated risk, and the adjusted risk ratios say very high there because we don't have any, as you can see, we don't have any events in the comparison interval, so we can't actually calculate a risk window, but we can calculate a lower bound of a 95% confidence interval. So you see those are generally speaking elevated with elevated excess cases. So that means excess cases in the risk period compared to the control period, or the risk interval compared to the control interval. So this is a similar table, but it's looking at 18 to 39-year-olds, both vaccines. So in ages in which both these vaccines were authorized or approved in the case of Pfizer. And again, similar findings. You see whether the vaccines are combined, both mRNA or separate analyses for Pfizer and Moderna. You look at the events in the risk interval or events in the comparison interval. You see the adjusted rate ratios there, all elevated, with the lower bounds of the 95% confidence interval above 1, and the excess cases in there per million doses administered. So if you look at the adjusted rate ratios for Moderna compared to Pfizer, regardless of the analysis by dose, you can see that they do appear elevated or higher for Moderna compared to Pfizer. However, this is not a direct comparison. So the take home there is there's an elevated risk for both Pfizer and Moderna. The individual analyses suggest Moderna is associated with more risk, but that was not a direct comparison. So the follow-up then is let's do a direct statistical comparison between Moderna and Pfizer, basically a head-to-head in these age groups for which you can actually do a direct comparison. So this is a graph which just shows the incidence of confirmed myocarditis or pericarditis in 18 to 39-year-olds, and just focus there on the left-hand side because that's the 0 to 7 day risk interval where we're seeing this clustering, and you can see, at least graphically, the incidence looks substantially higher for Moderna compared to Pfizer. Now, when you put that in a table, and these are multiple analyses, basically looking at myocarditis and pericarditis after day 0 to 7, after dose 2 in 18 to 39-year-olds, Moderna versus Pfizer. Whether you're including pericarditis cases or not, whether you're looking at both sexes or just males, the adjusted rate ratios are all elevated, some of these statistically significant, and are really in the range of about 2 to 3. So I think the take home from this is in the direct head-to-head comparison of Moderna versus Pfizer and VSD, we're seeing about a 2 to 3-fold increase risk for Moderna compared to Pfizer for myocarditis in the 18 to 39-year-old age group. So moving on to GBS, I'll go through this pretty quickly. This is from a presentation that FDA gave at their VRBPAC meeting. They looked at the VAERS reports and have observed substantial disproportionality with disproportionately more VAERS reports, more GBS VAERS reports submitted following Janssen compared to Pfizer or Moderna or their mRNA vaccines combined. The estimated observed to expected rate ratio when looking at Janssen compared to mRNA vaccines was 4.18. And based on this information, they added language to the EUA fact sheet on GBS. So this may be a little bit small, but this is from a preprint from a VSD analysis looking at GBS in the vaccine safety data link for Janssen, which is on the top there in red in the middle table, the table 3, versus, I'm sorry, yeah, it's Janssen versus mRNA vaccines combined there in the blue. And if you look at the unadjusted incidence rate per million, where I have them circled there in red and blue, you can see that regardless of the risk window or the Brighton level, the incidence rates are substantially higher for GBS following Janssen compared to mRNA vaccines combined. Actually, the difference is striking. And then we look on table 4 there on the bottom, you can see when looking in the 1 to 21 day risk interval or the 1 to 42 day risk interval, you're looking, this is Janssen compared to mRNA combined. The adjusted rate ratio is 20 in the 1 to 21 days or 11 in the 42 days. So a 20-fold increase or an 11-fold increase, depending on the risk window you're looking at for GBS, for Janssen, compared to mRNA vaccines combined. So I just basically gave you a summary of 4 adverse events for which we have sufficient evidence to establish a causal association or evidence that is suggestive, highly suggestive of a causal association, really in the case of GBS. I'm now going to wrap up with pregnancy outcomes. And I'll start off with some data from the vSafe pregnancy registry. And this was presented at a ACIP meeting. And just the take, they looked at risk of spontaneous abortion after COVID-19 vaccination. And I think that the take home here is the age standardized cumulative risk. And they had to do some of these adjustments because a lot of these women who enrolled early in vSafe were employed healthcare workers. So they tended to be women who may have been a little bit older than the general population because, again, largely employed working women in the healthcare field. The risk of SAB was 12.8%, and that's similar to previously published baseline estimates, which is in the range of 11 to 22%, which provided evidence that mRNA COVID-19 vaccination during pregnancy is not associated with spontaneous abortion. And then here's some early data on infant outcomes. I'm looking at preterm, small for gestational age, admitted to the NICU or neonatal infant death. And you can, the caveat here is that some of this vaccination occurred in different trimesters. So we didn't have sufficient data to really hone in on a specific trimester, but you can see that the percents of these outcomes here are within the range of what we would expect based on background, which is in the right-hand column. And then finally, this is a VSD analysis, which was a kind of a modified case control analysis looking at spontaneous abortion after mRNA COVID-19 vaccination. And it looks at adjusted odds ratios for receipt of COVID-19 vaccine within 28 days prior to a spontaneous abortion. And you see on the table here, when you look at the full population, or you're looking at gestational age by week when they were vaccinated, the adjusted odds ratios or by vaccine as well on the bottom are all around 1. So 1 is the null, no increased risk. And again, reassuring data that COVID-19 vaccination during pregnancy is not associated with spontaneous abortion. So I don't have a summary slide because I thought it was impossible to put this all in a summary, but I do want to acknowledge the contributions of the folks at CDC and also our partners in the Clinical Immunization Safety Assessment Project, the Vaccine Safety Data Link, and the FDA Center for Biologics Evaluation and Research. Thanks. Melinda, if we have time, I did have some extra slides on the myocarditis long-term follow-up, but I want to reserve enough time for questions.
So why don't you just do that quickly, and then we can move to questions.
So CDC is currently doing enhanced surveillance for myocarditis outcomes after mRNA COVID-19 vaccination of VAERS case reports. The reason we're doing this is our cardiologist consultants have cautioned us that although these cases do appear to be mild and people have symptomatically recovered and are doing well, you really need to assess these outcomes at 3 months or greater to assess the recovery status of the heart. So we're currently doing some enhanced surveillance where we're contacting these case reports and administering a 2-component survey. Number one, a patient survey to get at functional status, clinical symptoms, and quality of life, and also a healthcare provider survey. So contacting their cardiologist or whoever they're seeing as outpatient to do more of a technical survey. And this process is ongoing. And here's some references for that. So we're We currently have over 300 patients enrolled. The patient surveys are kind of outpacing the healthcare provider surveys. Number one, because they're given first, but number two, it is a little more difficult to get a hold of the healthcare providers and administer the survey. But this is a substantial cohort of vaccine-associated myocarditis case patients that we're following up on, primarily focused on individuals under 29, and we hope to have some preliminary data and be able to make that available in the coming months. Thanks.
Thanks so much, Tom. That was great. I really do appreciate both of those presentations. One thing that has struck me in this safety experience is how important the rapid information sharing has been. You know, with vaccines being used with the same vaccine products being used in multiple countries, or vaccine products using the same platform like the adenovirus vector vaccines, the fact that countries were able and willing to release information as pretty much as soon as they had it seems to have really been very impactful in safety monitoring globally. Is that something that either of you would like to comment on?
Sure, I'll start and then I'll hand it over to Tom. I agree completely. I mean, I think to me, this has been one of the successes. Typically public health tends to share with public health, but I would say in this response, in my experience, public health is sharing with public health as well as regulatory agencies. Regulatory agencies around the world are meeting regularly and sharing with each other. There's really not a lot of daylight between them. And regulatory agencies and public health are sharing with each other. I can speak from a GAC's perspective. The European Medicines Agencies was the regulatory authority for many of the EUL listed vaccines. And they participate. We have somebody from their committee who participates in our GAC's COVID-19 working group. And we do share confidential information. But, for instance, when TTS was going on, I know that FDA and the European Medicines Agency had all seen the data back and forth. I know that Tom presented to the EMA. I mean, there's really good, people are trusting confidential information sharing. And a lot of that is still going on now as we move into adolescence and boosters. All the different agencies want to learn from each other, and people are doing it and they're trusting each other. And I think to me that is a success. It's critical and it's happening. Tom, anything you want to add to that? I know you've been on the whole circuit, so.
I don't have much to add to that, but I do think there's been substantial information sharing between public health agencies, among regulatory agencies, and between public health and regulatory agencies, and it's really helped us both to make us aware of what's going on out there with our partners, but also getting early information that may guide what we do as well. And I mean, just for example, we happen to have quite a bit of preliminary data, on 5 to 11-year-olds, because we started our program pretty early. And for the past week, we have been basically presenting to multiple advisory committees in other countries on what we're seeing in 5 to 11-year-olds. And I haven't been on those calls, but I'm sure, I'm hopeful that that's helping guide public health decisions around vaccination. And we've had folks on our VAST call from international partners who have presented early data to us, which I think has been very helpful, either to help guide what we do or to confirm what we're seeing. And I think the international collaborations and also collaborations within the U.S. government have substantially advanced safety monitoring.
And I will also add that though a lot of the focus of what you've heard today is middle and high income countries, I would include that we are getting information sharing from low and middle income countries as well. We don't always have, they will share that with us confidentially. We don't always have the authority to then share that data publicly, which is why you may not hear it, but it is part of the story that we hear.
Yeah, thank you. Thanks to both of you for those comments. Another thing that seems to me to be different at this moment in time compared to, say, I don't know, the H1N1 pandemic is the wide dissemination of pre-publication reports. And we saw several examples in presentations today of reports that are available as preprints even before peer review. And that's a development that of course isn't related to the pandemic, but I think it really has facilitated sharing of information prior to publication in a way that has been, I think, pretty helpful. Okay, we've got a number of questions, and I will ask the presenters. The presenters can go on camera and unmute and ask their own question when I call on them, so we can see if we can make this work. So our first question in chat is from Rebecca Chandler, who has a question about signal confirmation by GACs. Can Rebecca go on camera and be unmuted?
Sure, thank you. Yeah, I had a question just related to the slide shown with the number of potential signals that the GACs had reviewed, and there was very few of them actually that were confirmed. And so I was just wondering what evidence is used to confirm the signal. For example, I was a bit surprised to say that Bell's palsy had not been confirmed when there was an obvious discrepancy even in the clinical trials. So I just wanted to know what evidence is discussed and is required to confirm a signal by the GACs. Thank you.
Thank you. And so I should also clarify that column isn't only GACS's, that is WHO's, their watch list that they maintain. So from a WHO standpoint, the things that are going to factor in are whether regulatory agencies have confirmed a causal association versus a warning versus something that they're continuing to watch. From a GAC standpoint, we really work by consensus. So WHO will bring a topic to us and present data from many of the places you've heard about today, as well as others that have confidential data that they're not willing to go public. We will discuss whether or not the data is strong enough that we feel we have to go out and make a statement about the data or not. You know, to me, whether it is quote unquote confirmed or a product warning is more of a regulatory one, but whether it is a signal that we feel merits public discussion is usually more done by consensus.
So those are, so the ones that are confirmed, so to say, are the ones that have had a GAC statement issued?
Not necessarily. There are some, for instance, the frail deaths in the frail elderly in Norway and the flu-like symptoms after the AstraZeneca. GAC put out a statement reassuring that we did not consider these a problem because we thought it was important for the immunization programs globally to have reassurance that we thought that they were okay, that these were not a new safety signal. Other examples are, for instance, for TTS, GACs has put out multiple statements, including at the very beginning before they knew whether there was an association, because we want to be open and transparent. You know, lack of information, as somebody in the chat noted, will get filled with a void with in this infodemic era. So the feeling is that you've got to tell people what you do and don't know. Some things are felt to be less of a public health concern than others initially, and so they may not rise to the level of a GAC statement. But if they were felt to be of a global public health concern, then WHO may ask GACs whether they want to make a statement, or GACs may say, we want to make a statement. We also try to do some coordination with regulatory authorities, so we're not getting either ahead of them or, ensuring that to the extent that we agree with each other, that our communications can go out together so that there's a unified voice. That doesn't mean that if we disagree, we would say that as well.
Thank you. There are a couple of questions in the chat about the safety data on 5 to 11-year-olds, since you mentioned that, Tom. Is there anything that you can share at this point with the group?
I think it's still pretty early, but the data we have right now, we're not seeing anything unusual or unexpected. We do have VAERS data, we have VSafe data. The reactogenicity profile in VSafe is consistent with what was observed in the clinical trials, and we're not seeing any unusual or unexpected patterns in VAERS. There are reports of myocarditis that we're in the process of evaluating, so I think it's still early, and we need to continue to follow up and evaluate that condition, which clearly is an adverse event of special interest. But right now, at least early, where we are right now, which is still pretty early, we're not seeing anything unusual or unexpected that would suggest a safety problem in that age group.
Great, thank you. I'd like to invite Nika Raffali to go on camera and unmute to ask a question about VAERS reporting.
Hi, can you hear me?
Yes, please go ahead.
Yeah, I was just curious when Tom was presenting the different formats for VAERS, whether you have a cell phone format, because here we have a lot of problems with continuity of power and, computers being fried and stolen. And so a lot of people do better with cell phone reporting. Can I also make a comment, Melinda?
Sure.
As the token South African on the call, I've got to say that we're all feeling pretty rueful about information sharing at the moment. Thanks.
Completely understandable. Thank you. Tom, go ahead.
I think the first question was, can you report to VAERS on a phone? Was that the question?
Yeah, I think that the example that you gave in your presentation, it looked like it was like on a desktop computer. Correct. And so in many parts of the world, people are more likely to be using smartphones. So is that a modality that's available in VAERS, I think is the question. Yes.
So we have as part of a modernization project, which fortunately happened before COVID, we developed a mobile phone friendly website or interface. So basically you get the same you get the same interface that you do and can report the same way you would on a laptop or a desktop, except the screen is sort of formatted for a mobile phone, right? So it doesn't look strange or distorted, and it basically has those nice features where it'll kind of run as you move through it. It'll pop up new information. So the answer is yes, we do have a mobile phone friendly interface to report to VAERS.
Great, thank you. Christoph Stefan had a question about death reporting in VAERS. Christoph, can you turn on your camera and unmute?
Can you hear me?
Yes.
And thanks for the presentation. I was just wondering, so I have two questions, but my question on VAERS was, what you hear a lot is that the reporting has gone up a lot with the rollout of the vaccines, and how do you, so I suspect that also the death reports have gone up. So my question is, how can you actually make sense of those death reports? Is there any ways I mean, if you look at the risk window versus outside the risk window, so is that something that is done also? I don't know. I didn't hear you touch on it. Sorry if I missed it.
So I'd say monitoring death reports and analyzing death reports in VAERS is challenging. I mean, it's spontaneous reporting. It's subject to biases, especially in the pandemic, because the FDA specified what was mandatory to report in the emergency use authorization. CDC put those requirements into the healthcare provider agreements that every healthcare provider or healthcare organization had to agree to participate in the program. CDC did a lot of education, outreach, and marketing of VAERS, and there's just a lot of awareness. So I guess it's not at all surprising to us that there's a lot of stimulated reporting and there's a lot more death reports. Also, as I'm sure you're aware, serious adverse events like death are more likely to be reported if they occur in close proximity to vaccination. So you've got a bias right there. I think really the best we can do in VAERS is to follow up on these reports, either by getting death certificates, medical records, or autopsy reports, and confirming the cause of death, and then looking at those causes of death compared to what we would expect based on vital statistics that's collected and published by our National Center for Health Statistics. And then being able to say, you know, what we're seeing in VAERS is not substantially different than what we're seeing for background rates of death. And the most common causes of death are where we know where we can establish cause of death in VAERS. It's consistent with the most common causes of death just from national vital statistics. That is problematic. Talking about death reports is challenging, and it's always difficult, it's difficult to talk about death reports in VAERS when you're seeing, more reports after COVID-19 than all vaccines combined. I will say the Vaccine Safety Data Link recently published a paper, which is a population-based analysis, which looked at the risk of death after Pfizer and Moderna and Janssen, and they actually found a lower risk of death after vaccination compared to a comparison group, which is not totally unexpected. That's likely the healthy vaccinee effect that we often observe, but we at least weren't seeing an increased risk of death. So I don't know, the best we can do is basically put these death reports and VAERS into the context of what we would expect to see in background, and also do additional analyses in population-based systems where we can actually try to establish risk, like we've done in VSD for COVID and for other vaccines like HPV vaccine.
And Christoph, did you have another question?
Unrelated, but yes, I was just curious about the biological mechanism around the myocarditis, whether we had any explanation. I think there was a question also, why is it more in male than female, and is there anything around what you read sometimes that there might be a specific. So I'm still unsure how the mRNA vaccines actually function in terms of which are the cells that express the protein actually, and could it be that it actually is being expressed in some of the myocardic cells, which would explain a direct toxicity rather than an immunological mechanism? Okay.
Go ahead, Melinda.
Well, I was going to say, I think those are really interesting and important questions, and I'm not sure we've got answers to them yet, although there's a lot of interest and discussion about that. But Tom, what's been the conversation within CISA?
Well, I think that the male to female predominance is basically following the normal epi of myocarditis. Is much more common in males than females. I guess I don't have a good answer to your question, other than what I think I can say is that mRNA vaccine associated myocarditis, the mechanism is probably different than smallpox associated myocarditis because the onsets are quite different. I mean, and for the mRNA vaccines, you're seeing this clear clustering 2 to 3 to 4 days after vaccination. And then for smallpox, it's in the range of a week or two. So it's probably it probably is a different mechanism than smallpox, but that's about all I can say. I don't know if there are others who may know more about the actual mechanistic evidence.
Okay, thanks. Thank you for those questions, Christoph, even if we couldn't answer the second one. Let's see. So Rebecca Chandler had a question about signal detection in the context of, I guess, misinformation. Rebecca, do you want to ask that question?
Yeah, I attended a webinar hosted by WHO and UMC earlier this week, where the concept note that was shared and the title of the meeting really related to, it seemed that the UMC and the WHO had concerns that that it was very difficult to find signals in the, I guess, in Vigibase due to concerns of an infodemic, that there was a lot of masking of signals, and I believe that there was even a concern that there were lots of false signals. And I was actually wanted to ask Tom if the U.S. had experienced, if they were really, I mean, we know that there are a lot of spontaneous reports coming into systems, but has it really been so problematic that it's been hard to detect meaningful signals for follow-up?
I'm probably not the best person to address that. It sounds like, are you saying is misinformation somehow distorting our ability to analyze data?
That was the kind of the concept of the webinar that was earlier this week about an infodemic masking, making it very difficult to see signals. And I was just wondering if the US had experienced that problem.
So I mean, I would say misinformation and disinformation probably makes our job in general more difficult, but I don't really know, or I don't have clear evidence that it impedes our ability to effectively monitor vaccines and detect and evaluate signals. That's about the best answer I can give you.
Yeah, and if I could just add to that, because I attended that webinar as well, and I think it's a great question. I think it's going to be a particular challenge in places who have, sort of early safety systems that are relying on passive surveillance that may or may not be consistently used. There are some countries that are concerned about reporting because they're worried that that will actually reduce the confidence in vaccines. Whereas in contrast, I think if absence of information gets filled in today's void, with people being very vocal. So I think, one of the things I think that helps countries like the US is the fact that it's not just the passive surveillance system. They've got these other, active surveillance systems, population-based surveillance systems, sentinel surveillance systems. And, this is where I put in the plug in the ideal world, in the same way that they were developing clinical trial sites for places so that they would be available in a pandemic. Now I give India, I applaud them in setting up all these systems now. In the ideal world, we want countries to be able to have these systems in place prior to the pandemic. So I think this is a particular problem in places that don't have the infrastructure or don't have the communications, even if they have their data, don't have the communication systems up and running to be able to get good information out rapidly, because somebody else will fill the void if you can't do that.
Thank you. There's a question about adverse events following booster doses. Is Margarita Greve-Eastal, would you like to unmute and ask your question?
Sure, thanks, and thanks for a very good presentation from both of you. I was wondering, since it seems that the myocarditis signal could be dose dependent, since we see it more often after the second dose, and I guess that's also quite a new experience regarding vaccines, except for anaphylaxis. If you have any thoughts on what we can expect for the booster dose, especially in the younger population, now that we're rolling out booster doses in a lot of high-income countries. Thanks.
I think it's still pretty early in the booster dose program, at least in the United States, and there probably isn't that there isn't that much information in these younger ages, like individuals 18 to 24. But the data we do have, and I'm specifically talking about myocarditis, we don't have any evidence so far of a safety problem for myocarditis after booster doses with respect to what we know about second dose, if that makes sense. I mean, we know there's an increased risk of myocarditis following second dose, at least in certain populations, mainly young males. But we're not, with respect to booster doses, we're not seeing anything concerning using that second dose as sort of the anchor. I will say we do have some reactogenicity. We have some pretty good reactogenicity data after third dose, and it looks like regardless of the product, the basic local systemic reactogenicity and health impact is lower after third dose compared to second dose. I guess you can speculate about the reasons for that. Maybe it's a longer interval, but it appears to be lower for booster doses.
And just to add, Israel probably has, because they started their booster doses earlier than us, probably has the most data on that. They presented to FDA's VRBPAC when they were about halfway through the monitoring time for myocarditis in the young adults. And there was certainly not, it was less than what they had seen after the second dose at that time, but they had not completed their interval. But they've been pretty open. If I haven't seen the follow-up data, and I don't know if Tom or Melinda, if anybody else has, but I think that if it had elevated, we would have seen it. More generally, in terms of boosting, there was a SAGE meeting yesterday, and there was a presentation on some of the data on heterologous boosting. And there's a little more reactogenicity with some series versus others. There's a paper that came out in the Lancet recently by Munro, M-U-N-R-O et al., done in the UK, that looked at a whole variety of vaccines. Sample sizes are pretty modest for these. They're usually 50 to 100, but it can give you a sense of the common reactogenicity profile. I think, as Tom said, for the ones we've seen in the US, it's been less. For some of the sequences they've done, it's been slightly more reactogenicity, but no safety concerns that were identified. But I think Israel probably has the best myocarditis data to date. Melinda, I don't know if you've seen more recent data from them.
What I've seen is consistent with what you said, that at least the myocarditis rates seem to be lower after the booster dose than they were after the second dose. And I think we're out of time. So
can I just add one last question, because I guess also for the ones who have had myocarditis after the second dose, you would not recommend a third dose, I assume.
Oh, Tom.
I'm not sure what the clinical we have guidance on our clinical considerations website. I'm not sure specifically what those recommendations are. I'd have to check what those are. I don't think it's just a necessarily you should never give it, but I do think there may be some. I mean, it may be more nuanced.
It's a little more nuanced, and part of it will also depend if there's availability of other vaccines. they want to see people are recovered from myocarditis before they do anything. But there is a little bit of nuance to the recommendations there.
Okay, well, thank you so much. Thank you for those questions. Thanks especially to our speakers. Those were great presentations, very comprehensive. Appreciate everyone's attendance and all of the questions. And Philippe, do you have any closing comments?
No, I thank you very much, Melinda. Thank you for your great sharing. That was appreciated, and these very insightful presentations we had. Thanks to all the participants. I know that several of the participants are interested with accessing the mortality risk paper that Tom mentioned. So assumingly, I hope it's an open access paper. If so, if you could share with us, we could post on the Alumni website, or if you could provide the link, that I think would be useful. We got several of the participants requesting that. We also had several participants which questions were not directly answered, but I think they were actually covered through the additional discussions and through the presentations. So thank you, everybody, and I want to thank you, Carol and Francois from Outcast, also for the help with the organization and the support of the orchestration of this meeting. And I wish all of you a good holiday season, and hopefully less COVID at the turn of the year. I think all of you could do with some rest and being a bit off the hook, but we'll see how things go. So thank you very much for this webinar and your participation. And we will, of course, post the recording on the website. Usually these webinars are attended by quite a number of participants, but they are actually watched by thousands more of interested parties, that they be ADVAC alumni or interested parties from other courses or other organizations, since we post these with actually open access. So thank you again, everybody. Bye-bye.